All right. Good afternoon. I would like to thank organizers for inviting me to tell you about our recent work on extracellular space and seizures. So first, I would like to acknowledge contribution of people to this work, which got published in JPhysiol last year. So when I'm going to say I really mean them, because it's mostly them who did the work. I would also like to acknowledge my collaborators at Downstate, Catherine Perkins, Doug Lynn, and Hilary Michelson, and also the funding from NINDS from New York State. That was in collaboration with Jeff Goodman, and then funding as a C award from the Office of SVPR at SUNY Downstate. So, as we already heard a couple times today, approximately 30% of people with epilepsy continue to have seizures despite intensive treatment with available anti-seizure medications. So what this is telling us that we need to do better. We need to find new treatments, and essentially one way how to go about it is to look at some pathophysiological processes happening during the seizures, which hasn't been really studied before, and see whether blocking these mechanisms can lead to new therapeutic approaches. So in my lab, we are focusing on brain extracellular space. And recently, we found a new phenomenon which is happening in brain extracellular space during the seizure. And that's what I'm going to tell you about. But before I go into the data, let me tell you a little bit about brain extracellular space. So you can see here on this drawing that obviously in brain we have neurons and we have glia, the main two cell types, and these cells are surrounded by the extracellular space. It's been also known that brain extracellular space and changes of its volume can influence neuronal activity. So how is that happening? It is happening because in the extracellular space we have ions. We also have neurotransmitter and neuromodulators. And when the volume of the extracellular space gets smaller, the concentration of these substances get enhanced, and that can lead to higher activation of neurons. In addition, there are factors like ephaptic interaction. These are effects of electrical fields of one neuron to another. And when extracellular space gets smaller, it means that these neurons get closer to each other. And these effects get also enhanced. So the phenomenon which we discovered in my lab is called rapid volume pulsation. This occurs during epileptiform activity, and essentially it is rapid shrinkage of extracellular space, which happens with every discharge of epileptiform activity. So first, let me just briefly tell you how we studied that, how we do the measurements. We use a method which is called probe transient quantification method. And essentially the way how it works is that we take a probe molecule, a small cation tetramethylammonium, we add it to the brain tissue and we let it to distribute in brain extracellular space. That's shown here by the pink color. And then we have a probe which can sense and quantify the concentration of this probe molecule. And this probe, this sensor, is ion-selective microelectrode, which has two compartments. One is for measuring the concentration of tetramethylammonium, and the second one is for measuring the field potentials, electrical activity in brain. And now when we take this method and we induce epileptiform activity in the brain tissue, in slice or in vivo, this is what we start to see. Here in the blue color, we see the epileptiform activity induced by our chemoconvulsant. And with each discharge, we see a signal on the other barrel. The concentration of tetramethylammonium is changing. When the discharge happens, the concentration rapidly increases, and then gradually comes back to the baseline. And then next discharge comes and it repeats. And this goes over and over again. So what is our interpretation? Our interpretation of this phenomenon is that after the baseline, when the spike comes, we get rapid swelling of the cells and shrinkage of the extracellular space, and this leads to the increased concentration of TMA in extracellular space, and then gradually the cells recover back to their normal volume, and so does extracellular space, and TMA concentration comes back to the baseline. And we call these events rapid volume pulsations. We observe them in multiple models of epileptiform activity, most of them induced by chemoconvulsions, but we also saw it in a model of Has3 knockout model of epileptiform activity, which is spontaneous epileptiform seizures. We found that there is a relationship between LFP discharges and RVP 1-to-1, that these events last as long as we are willing to measure for many, many hours. And through the quantification, we found that the extracellular space at the peak of the event shrinks by approximately 15%. So our next question was to try to figure out what is the underlying mechanism for these rapid volume pulsations. And the reason for trying to figure it out, if we can figure out which channel or transporter is responsible for creating these rapid volume pulsations, maybe by blocking it, we can stop rapid volume pulsations, but also the epileptiform activity. And we, by me, Robert, tested a number of channels and transporters, both on neurons and on astrocytes, and essentially, to cut the long story short, he found only one, which was very effective. And this is electrogenic sodium bicarbonate cotransporter, NBCE1. Great. And we studied this using DITS, which is an inhibitor of this transporter, and what we found was that when we applied DITS on the brain tissue where we previously induced epileptiform activity, the RVPs and also epileptiform activity disappeared. So to summarize, what I told you so far is that we found rapid volume pulsations during epileptiform activity, and they decrease extracellular space. We also found that the blocker of NBCE1 transporter stopped RVP and epileptiform activity. So we hope that RVP and NBCE1 could be new potential targets for treatment in epilepsy. And our future directions is to figure out whether RVP is present in chronic animal models of epilepsy. We also would like to determine which cells swell and whether RVP is present in human epileptic brain. And I want to conclude by one experiment, which we did in a collaboration with Doug Lynn's lab. And this is, we look for the RVP, whether they are present in post-traumatic epilepsy. So in this experiment, we use CCI model, cortical impact model, in rats. And then we make slices about 3 to 4 weeks later. And in this preliminary experiment, we found that RVP are indeed present in there, and we are very excited about this finding. Thank you for your attention.